This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 131, recorded April 29th, 2011. Hi, everybody. I'm Vincent Racaniello, and it's time to talk about viruses. Joining me today, right here at the TWiV studio, Dickson Despommier.
Hey, Vince.
How you doing, Dickson?
Doing very well, thanks.
Really well?
Really well.
I'm glad to see you again.
Oh, glad to be
Your voice is a little bit low. Let's crank you up here.
Crank me up.
Because you tend to be shy, Dickson.
Right.
You're retired, but not retiring, right? Is that what you had to say?
I've always been shy and retired.
Also joining us today from Western Massachusetts, it must be Alan Dove.
Good to be here.
How are you, Alan?
Doing okay.
I bet you have cloudy skies with a little blue peeking through, right?
Actually, we have blue skies with little puffs of cloud passing by, and it is an absolutely glorious day for the first time in I don't know how long.
Exactly.
So this is what passes for summer here.
The floods have receded where we are. So.
It'll last a week, and then you'll have winter, right?
No. Then we'll have road repair season.
That's right. Exactly.
Rich Condit isn't with us today. He is at the Poxvirus Invitational Golf Tournament, which is in Gainesville. This time of year, all the pox virologists gather to play golf. Instead of pushing large viruses around, they push around small golf balls. But we have a special guest today we're really happy to have.
You might say they're pushing a different envelope.
Oh,
Alan, that was just for Alan.
I'm really happy to welcome Dr. Brad Thompson, the Chief Executive Officer of Oncolytics Biotech. Welcome to TWiV, Brad.
Oh, thanks very much.
You're here because one of our listeners, Bill, is an aficionado of using viruses to treat cancers. And he's been asking us, I think almost 2 years now. And so finally, we've come around to have you on. And we're going to talk about your company and how it uses reoviruses to treat tumors. But before we do that, I'd like to find out a little bit about you. You have a PhD. Tell us just briefly your background, how you came to be at Oncolytics Biotech.
Well, undergraduate and PhD in microbiology, so classical microbiology, which I still have a great fondness for. And then I came back to Alberta, and like many good Albertan kids, worked in the oil industry for a while. And it's kind of unless you do that, you're not really socially acceptable here. So, but honestly, after a couple of years of that, I was just like, I have to get back to something interesting, and I apologize to any rock geologists that are out there that I just offended them. But the oil industry is very different than what I'm used to, which is more discovery based fun stuff. And from there, I went and did a lot of fermentation research, and then toll manufacturing and scale up work in biologics. And then founded a gastroenterology company, which was spot on with my expertise. We were doing a number of different infectious diseases, hospital-acquired infectious diseases that are GI related. And then I had this nice opportunity to come along to work in this particular company. I was literally sitting in my office one day, and a colleague of mine in the government phoned me up and said, "There's this very interesting research group at the University of Calgary. You're a microbiologist, so you will understand half of what they're about to say to you. And they're looking at developing an interesting new viral vector for treating cancer." And normally I would have probably tried to find an excuse to not talk to another university group about something I didn't know about. But I just had melanoma myself, and my mom had just died of adenocarcinoma in the lung, and my favorite uncle had just died of head and neck cancer, coincidentally. And all in about a 6-week period. Wow. And so they caught me at a moment when my sort of awareness of oncology was a little higher than it had been. And I do know microbiology, so that's a good thing. And so they came over and gave me a visit, and we talked for probably 6 or 7 hours on that first visit. Wow. And they showed me just animal model after animal model that they'd done. And they were all good, solid. I mean, it was not jury-rigged animal models. They were good, solid, relevant animal models, which quite surprised me. And I was just intrigued, just the thought that you could actually do something in this particular area for something that meant to me something personal at that point. Kind of caught my attention. So, and I went home to my wife. We were late in 2 phase 3 studies in my previous company. And I said, hey, I'm going to quit this and go to a preclinical thing again. And I'll be paying myself $1 a month to start because we don't have any money. And she just shrugged and looked at me and went, hey, great, good for you. And
right.
Fortunately, she's has been the traditional breadwinner in our family. So I get,
she's still in the oil industry, I take it.
She's an investor, so
right.
and she does a lot of energy investing. So I had a bit of latitude there from a personal side to actually to do those kind of things. But just the whole this whole area just really grabbed my attention. And then I started digging into it, and all of a sudden I find out there's all these people that I had known when I was, in school and whatever, who had all hopped over kind of to the dark side, as people would think of it. And we're looking at these different viral vectors early on. I mean, Grant McFadden, who's been on the show, was a postdoc down the hallway from me when I was doing my PhD, and I mean, it's really it's a pretty tight knit community, honestly. And it was just kind of interesting. And when we started looking into it in more detail, I mean, there was absolutely no regulatory pathway. Nobody knew what the real technical questions were. Nobody knew where we were going to trip up, and which is exciting. I mean, that's the fun part of doing anything is being kind of first, or amongst a group of people being first. And here we fast forward 11 years later, 12 years later now almost. We're in a phase 3 that's going to be in 11 or 12 countries in a couple of months.
Wow.
I've got a couple of other randomized studies running at phase 2. I'll have another 2 running by the fall. So by this fall, we'll have 5 coincidental randomized studies and different indications running. It's just a you think of that whole path from day 0 to here, and it's been exciting. It's been fun. It's been challenging, but it's something that I'm really, honestly, seriously, really glad that I did. And
so when we told Rich you were going to be on, he mentioned it to Grant, and I guess you and Grant knew each other at some point in the past, right?
Yeah, he was a pure virologist working down the hallway with our favorite pox virus, vaccinia, and as a postdoc, and it was just before he actually. He was just finishing off his postdoc when I was just finishing off my PhD. My PhD is more bacterial orientated. Mm-hmm. I was on the other, the opposing camp down the hallway. He was at the virus end, and I was in the bacterial infection.
And now you're in the viral. You're in the viral end now.
Yeah, well, I'm quite happily in the viral end. I've actually done a lot more viral work than I have bacterial. Viruses are. When you look at. And this area is, I think, just a good example of the elements of virology that you can use. I mean, yeah, you have all these different viruses. And everybody always says because it's a virus, it must be the same, which couldn't be further from the truth, which we all know on this line. But they're different sizes, they're different immune presentations, they have different mechanisms of action, different replication cycles, et cetera, et cetera, et cetera, and different tropisms for tissue. You have all these different things, and that's really, I think, a unique element of this particular area is that we're going to come up with a suite of therapeutics that includes different viral vectors, different engineering tricks that people have done with them. And in the end, I think we'll end up with a suite of products that address a lot of different cancers. No one of these products is going to address everything. And I think you just exploit the things that we've learned along the way as a group and apply that to we'll be able to almost tailor what we're looking at in a few years. I mean, who'd have thought size would have mattered so much when you're talking, ranges of, 20 to 200 nanometers. Well, it does. And who would have thought that immune response could completely block one virus and be completely invisible to another virus getting into a tumor? No, I mean, not just to the tumor, but into the tumor. I mean, all these things we didn't know. And that's all been, we've all kind of learned from each other. In the early days, we had Onyx with 015 was really kind of trail breaking a lot.
That was an adenovirus, right?
It was adenovirus construct, and they just had a horrible time making it. I mean, who thinks about manufacturing issues and these things? Terrible time making it. But do you take a look at the thing? They'd made, 3 genetic changes? So you lose about 90% of productivity with every genetic change, as it turns out. And, 3 leads you to something that's not very good. 1,000th isn't very good. And very early on, Onyx was flagging to the rest of us who were way behind them at that time. Pay attention to manufacturing. Some of us have. There's been a few of the companies that have really paid attention to it, and it's paid off hugely. And I mean, you have processes that when you're running into late stage are rationalized, they're cost effective, they're consistent, they're everything that you need for a biologic. But it was again learning from our colleagues. I think Oncolytics is probably in 10 years from now, when people look back at what our contribution to that path will be, is that we were, to a greater extent than I think anybody else, really looking at combination therapies and their effect on tumor permeability to the virus. Who would have thought that would have been an issue? If you infect the surface of a tumor, it should be no problem. Well, it is a problem. And we found that, certain agents really increase tumor permeability to viruses. And, taxanes, that's a universal fail-safe for us. Radiation, almost universal for us. And not in humans, but in animals, the whole VEGF family, VEGF itself, Avastin, Sutent, that whole family does that quite nicely. And other people are emulating that now. You're starting to see that show up in their programs. And so everybody has been contributing little bits and pieces along the way. And and all of a sudden you have this field that has our colleagues at BioVex, who are now part of Amgen, have two phase 3s under SPAs, which are fairly late on. We've got a phase 3 in different indication and under a SPA, and a bunch of phase 2s in behind that. We've got pox viruses in phase 2s. We've got measles virus in phase 2s. We've got a whole group of people working on different elements that have learned from each other. And I think that's the key thing. Now, this collegiality would completely disappear as soon as we start getting competitive. Like poof,
Sure.
But for now, we have every 2 years, we have this virus meeting. We just had it a couple of months ago in Vegas. And it's, virus oncology. And you have like 300 or 400 people who this is all we do. And it's collegial. It's been just remarkable how in the last, if you go back 4 meetings ago, so go back 8 years, everybody was doing early stage phase 1s and retinkering and playing this and playing that. And now, we have a panel at this time with 3 or 4 companies who are probably going to be filing for product approval within 2 years. That's just an amazing change for a whole sector. But that's
actually fairly typical for biotech fields. If you look at any of the major therapies that have come out of biotechnology, any of the biologics, I've been following this for a decade and a half now, and you always see this initial group of companies, a lot of small startups, and a very collegial atmosphere, and everybody figuring out what mistakes to make so they can learn from them. And then eventually there will be, as you have already predicted, there will be a change in tone, but that'll come later as the approvals roll in.
Oh, absolutely. And I think a bit of the first change in tone is really Amgen buying BioVex. All of a sudden, it's not a number of our small players kind of hanging out with each other. All of a sudden, we have a very large player who's now integrating, phagocytizing BioVex. And
exactly. Engulf and devour, I think, as I put it.
I think some of the BioVex guys actually view it that way too, and
right,
as
right.
probably what Amgen's a great company.
So are we talking about curing or just holding at bay these tumors that you're going to be talking about?
I think it's going to be a mix. And of course, we get into the argument about when you get rid of a tumor and it recurs, whether it's the same tumor or whether you're just getting reseeding from somewhere else. But I think there's certain tumor types, certainly that we've seen complete responses, and that are durable for the period of time that you've seen. And certainly more so in metastatic lesions for us in particular. I mean, our REO requires an activated RAS pathway, which has a much higher incidence rate in metastatic disease. And of course, Mets are likely to be clonal expansions of single or a couple of cells stuck together. So they're much more consistent genetically. And so if you get REO into a metastatic lesion that has a RAS pathway, it'll completely clean it out. I mean, and it doesn't recur. Where you get into, I think, more issues about control or getting into, like partial responses that last while you're treating and then kind of spring back after you quit treating. The primaries seem to be kind of more in that category. Occasionally you get CRs in primaries, but you're more often getting nice aggressive PRs, but still PRs. And you're going to buy people lifespan with that, and you're going to do some good things for them. But the tumor architecture, I think, is critical in primaries. Sure. You get all these kind of internal barriers and mixed strata, healthy and non-health and neoplastic tissue. And but I think it depends on what you're looking at. And
I remember the original work of Judah Volkmann's group up at Harvard, always said that you could cause a tumor to regress right back to the original cell that caused it, but the original cell took advantage of existing circulation. So the approach to limiting the tumor by inhibiting the growth of capillaries only worked for new capillaries and not for the old one that was already there. So it would just grow right back again. And I don't know how far that's advanced since he's passed away, unfortunately. But that was the complaint, that these are great therapies while you're using them, and the moment you stop, everything starts to go back to the way it was.
Well, I think one of the mistakes that people make in oncology frequently when they're evaluating it is they'll say, well, this therapy didn't cure the cancer, therefore it's no good. But if you look at the reality of the way tumors are treated in the clinic, it's seldom just one approach that's being applied.
Oh, sure.
Yeah, no, it's a sequence of actions that does sometimes lead to quote unquote a cure, right? And we see that, like head and neck cancers, I think, are a really good example of that. I mean, the first time you get head and neck cancer, if they could just do surgery, they will, and they'll combine it with radiation, and half the time you're cured, you're fine. It works great. The other half the time, life isn't so good, and it's usually when it metastasizes. And but yeah, I mean the cure for cancer, I think, has not, that expression has not done our sort of research and industry a service.
Let's talk a little bit about reovirus. It's one of your main oncolytic agents, I presume. And tell us, our listeners are a pretty broad audience. Tell us what it is and why you are using reovirus in a lot of your studies.
Well, Reo was discovered in 58 or 59 in the feces of a small child, clinically, and as typical in that era, the golden era of microbiology, people would then go to try to figure out what it did, and you didn't have to wait for 5 years to get a permit to grow it up in the lab. I mean, they went straight down the hallway and threw it on some cells and grew up some, and then started doing things with it. And after 4 or 5 years of failing to find out what Reo did, they ordered up a batch of prisoners in the South, like they used to, and treated 27 quote volunteers with pretty high dosages intranasally of Reo, and no statistical correlation with anything. And
tell us what kind of virus it is.
It's a double-stranded RNA virus, okay. and it's a relative of Rota, which is why it was interesting.
All right.
Really,
sure.
So it's a kind of non-pathogenic relative of Rota, is the way I think of it. And so people just lost interest in it. I mean, and so they started doing research on it because it was a relative of Rota, but wasn't pathogenic. So the joke that if a grad student, you found a grad student mouth pipetting it, you didn't have to throw in a containment suit kind of stuff. And so that went on for about 20, 25 years, and people learned a lot about this interesting, durable little double-stranded RNA virus. It was easy to grow, which was good for labs. It was very durable, so you could make up a batch, and 6 months later, people could be still, pipetting out of the common batch in the lab. So
sure.
People like that. And labs, who wants to be spending time making something when you're investigating it? You want to research it. I mean, all those practical things. It's a good research tool.
Why is it called Reo, by the way?
Respiratory enteric orphan virus. So it
wasn't found in Rio, South America.
No, it's REO. Yeah.
but that's
just
really
a play on words. You could have just. Sorry about that.
I think that was coined by Albert Sabin too, right?
I believe it was actually.
Huh.
And it's its natural habitat is the lining of the lung and the bowels, but it doesn't cause a pathology, and of course that I asked that question pretty early is why. I mean, and
Sure.
I haven't really found a lot of things in my experience that affect the lining of your lung and bowel that don't cause something. A cough, diarrhea,
That's right.
bloody diarrhea. My previous history, anything to do with the GI tract, I always go right straight to the bloody diarrhea thing. But
but that's partly because we've noticed the things that cause disease.
That's right, and we ignore everything else. But when you look at, you actually sit down and you look at where reovirus localizes, and the bowel's the primary natural habitat, and it's a pan-mammalian virus, so which helps. So the virus, it's a fecal-oral transmission, so fecally contaminated water or food, which unfortunately is all food. Water almost. You get a low level of infection in the crypt cells in the bowel, and you go, well, why the crypt cells? And then you look at it, and those cells are bathed in EGF. And then you say, well, what does EGF do to a cell that makes Reo infected at a low level? And of course, EGF, and we know now, drives the RAS pathway.
Uh-huh.
And so if you take any cell basically and stick it in a bath of EGF, you will drive it into a low level of RAS activation, which is what Reo needs to give a productive infection. And so you have this one cell class, it's the same in the lungs, that the virus will grow in at very low levels, fecal oral into the bowel, replicates, passed out in the feces, reinfection. And that's a cycle. The reason you don't have a pathology is that it takes Reo about 3 days to kill a cell, and those cells are actually forced into an apoptotic death, and not coincidentally, because I don't believe coincidences in nature, about 3 days. So they kill the cells at the same time they're dying anyway. So there's no pathology.
So is that why people don't raise neutralizing antibodies against it, or do they?
They're very, very low. It's a mucosal immunity. The virus doesn't actually cross the gut barrier or the lung barrier. And so you get a very, very low level, very transient, as they always are, mucosal immunity. And so when you look at humans, if you're looking at the old doubling scale, which unfortunately when you date my lab history back is when we did antibody titers, that's how we did them. You're looking at titers of 2 or 4, 8, 16 normally in a patient population, but it's almost 100% of them. Like virtually everybody has a very, very low level. Now, when we treat people systemically with Reo for 5 days in a row, you can drive those antibody levels up to 20,000.
So this is good because you're using it as a therapy, so that it doesn't cause pathology. So you can actually say that you're not going to do harm. At least you might not do good, but you won't do any harm by giving this virus along with what you're trying to.
Yeah, I mean we're just under 500 patients when you.
It's very cool.
Have our patient population, which is about 400 in the NCI, is tacked on, and Reo as a monotherapy on a 5-day cycle will give you in patients that are likely to have a tumor response, and that's key. Low-grade fever, degree and a half fever, low-grade muscle and joint pain, and low-grade fatigue, and low-grade neutropenia and lymphopenia, which you'd expect on day 2 of a 5-day cycle or day 3, and it goes away the same day.
Interesting.
And we can mask all those symptoms with Tylenol, paracetamol in Europe, and Advil.
So in tumors that are having a low expression of RAS, is there a way to ramp that up to make it even more susceptible?
Reovirus actually does that. That's actually going to be published later this year. Reovirus actually drives the expression of EGFR. So it's
I'll be darned.
It's a tricksy little fellow.
EGF, epidermal growth factor, binds its receptor EGFR, and this drives cells to undergo mitosis, right?
Yeah.
And the RAS pathway is the signaling pathway that's signaling from the receptor binding all the way into the nucleus, making genes that are needed to get the cells divided. So what you're saying is that reovirus prefers cells in which that pathway is activated, in which to replicate, right?
Well, and the touch point is
conveniently a lot of tumors have activated the RAS pathway.
Right? Absolutely,
right.
I mean, cells have that lovely innate double-stranded RNA defense mechanisms that's not immune, that's mediated by PKR, and PKR binds to unique viral sequences on double-stranded RNA and just shuts down. You don't get protein translation even, so the virus can't replicate. But when the RAS pathway is constitutively activated, it actually deactivates PKR. So basically, you just have a permissive cell environment for viral growth.
For non-viral speakers, PKR stands for what? Protein?
Phosphokinase R.
Okay.
And it's activated by double-stranded RNA. when viruses infect cells as part of the innate defense system.
So I think we should pause just one moment here and reemphasize the fact that if you're going to be a virologist, you better know your cell biology.
And unfortunately, when we're talking with a group of people who, as we all are on this thing where we all kind of have done this before, we all retreat into a foreign language awfully.
Exactly. Sure. Every group does it. It's all jargonized,
but we actually have a little reminder at the top of the show notes about that we're talking to everybody.
Talk
here.
plain speak, and I agree.
That's right.
So to strip the science away, basically all you're doing is taking a cell that normally has a defense mechanism against this type of virus and deactivating it so the virus can replicate and kill it in a few days. So this is a fully replicative virus. You get about 40,000 progeny virus per cell infected. And so you just need to infect part of the tumor, and then the virus will replicate and spread through the rest of the tumor. Now, of course, that would just be a curiosity if it wasn't associated with some kind of disease, right? And we could have found all that biology out just by looking at the lung and the bowel, and then everybody would have went, oh, that's neat. That's very interesting. And that would have been the end of it again. But, oncology is certainly the area where you have the most sort of RAS pathway activity. I mean, you're looking at about probably 2/3 to 75%. Any primary carcinoma has a RAS pathway involvement. A lot of lymphomas do, and most metastatic disease. So basically you have a genetic predisposition from a family of things, because it's not just EGFR, it's anything on the RAS pathway will do the same thing. RAS protein itself, RAF kinase, everything on that path.
So why is there colon cancer then?
Well, we've actually treated polyps with Reo. If you give animals, push them into polyps and then give it to them orally, you can clean up polyps quite nicely with Reo. I think it's dosage and frequency of exposure. And of course, there's a lot of colon cancers that are probably not polyp related. But so I would expect that we might have a higher level of colon cancer than we do now.
And then it doesn't breach the epithelial lining, so you wouldn't get it to the primary tumor then.
That's right. If it penetrates the wall, it's too late. So the coincidental infection with Reo, because it's very transient, a couple of weeks after you've been infected with Reo, and the wild infection cycle, it's gone. It's completely cleaned out. And
I was looking for a positive role for this in cancer monitoring. It would be great if you could prevent bowel cancers like that, but I guess.
Well, I mean, I would drink this stuff regularly for bowel cancer, but I think I probably, the H. pylori story comes to mind when you're thinking about doing stuff like that.
That's right.
Which is a really
great. Now a whole industry of alternative health is going to spring up selling canned reovirus.
That's right. We had that run on tapeworm segments for a while back in the 1920s, but that didn't work either.
Well, the tapeworm is making a comeback. So that's
well, actually, the roundworms, not the tapeworms, but the roundworms.
Oh yeah, roundworms for Crohn's, right? So
that's right. We can cure Crohn's disease now.
So starting with the observation that this virus likes cells in which RAS is activated, and then knowing that a lot of tumors have this activated, what did you have to do to reach the next step then to say that this might be feasible for using in people?
Well, you just went down the hall and found a few prisoners, right?
No, that was Dr. Sabin in 1963.
Yeah, a few former hockey players would have done just well.
Yeah, things that people wouldn't have missed. Absolutely, or grad students or med students or law students. The typical profile for clinical trials. No, I mean it was a pretty serious issue to say, listen, it's safe in the natural infection cycle where you're taking maybe 100 viral particles and swallowing it in your food by accident, and being infected in something that doesn't transfer into your body anywhere, to leaping to injecting now 3 times 10 to the 10th active particles a day intravenously for a week on end every month. That's a big jump. And so you had to get through all the manufacturing stuff. I mean, this used to be made on people used to grow them on animal cells, of course, and so we had to get them on human cells because nobody was going to let us, treat people with animal cell derived product, even back when we started doing this. So you had to go through that whole process. Tons of toxicology. Like we just did a massive number of animal toxicology studies with a lot of primates. I think we ended up doing 15 formal animal toxicology studies, and looked at cardiac function in primates and just really extensive.
What was your big animal model tumor that you used as the proof of concept?
Well, most of the animal model work for what I think of as unnatural animal models is all rodent, and so you're looking a little in the melanoma models. We did do some naturally occurring tumors in canines early on too, mostly sarcoma based, which is the most common type of tumor that you see in canines. And everything was fine. I mean, everything was safe. Now it's kind of, we discovered if you don't have tumors, you don't get side effects with Reo. That kind of makes sense in hindsight. If you give somebody a big, huge dose of this and they don't have a tumor for the virus to grow in, you look for the virus, it's gone in half an hour. It's completely cleared. So it's not such a big deal. Once the virus inhabits a tumor, then you get active viremias, which is virus in the bloodstream, and those viremias can go on for several weeks, and that's the start of the viremia is when you get that minor side effect profile that we saw. But of course, we didn't know that when we started. So we started out like everybody else because the agencies just tried to do this by doing intratumoral. Now, nobody in those days knew that the tumors shed virus. We thought they did. Certainly the animals they did. But the agencies thought that was safer. So our first 3 studies were intratumoral. And so we did an all-comers study.
These are in mice, you mean, right?
No, in people.
This is in people. So we're past the animals already.
Yeah. Our first clinical studies was in all comers in people. We did dose escalation in tumors. Then we did a small T2 prostate study. So prostate cancer hasn't metastasized by either the lobe or the prostate gland. And then, which I think is, and then finally, I think the penultimate from a safety perspective, which was a glio study, brain cancer. Because if you're going to see clinical symptoms of inflammation and swelling, the brain is the best place to do it. I mean, it's exquisitely sensitive from a clinical perspective at showing off side effects you're not interested in. You can suffer a lot of things in normal tissue that your brain won't put up with. So that was kind of the acid test toxicology study from my perspective. So we got through that and we kind of went, okay, that's great. It's in those intratumoral things. We have to make the leap to systemic, and that's when we ended up having to do all that primate work. I mean, we hadn't done the primate work until then, and so the agency said, go back in and do massive long-term exposure in primates. Daily infusions for 6 months with monkeys, monitor cardiac function, monitor this.
Good heavens!
And each one of those monkeys costs about the same price as about 4 clinical patients in a clinical study. I mean, it's a very expensive program, and it's and we knew unnecessary, but you do it anyway. And so we've got through all that. That was all fine. And the agency then smiled at us and went, "You do realize you're not going to see any side effects unless you put tumors on those animals." Thanks, guys. So we did a tumor-bearing animals, toxicology, and then that's when you see the low-grade tumor. We did it in rats actually. You just put the sensor chip in the rat, and you put it over a scanner to take its body heat every day, and things like that. And then we did IV, and we did an IV monotherapy study in the UK and one in the US, just to make sure we weren't getting jurisdictional bias. And then that just opened the door. So then, once we got that monotherapy systemic behind us, that just opened the door, and then we started doing all the co-therapy work and started exploring it, and that's when our enrollment exploded. And but we're still obsessive about following safety. What I find interesting, this whole field, if you want, I'll put on my whole field hat for a second here. You take all the gene therapy studies, it doesn't matter whether it's oncology or not, anything that's used a virus vector, and all the oncology stuff, you're probably having about, it's probably about 10 to 15,000 patients now, if you look worldwide. And we've had, probably less than 10 deaths that you could associate directly with whatever virus that's got the lowest side effect profile of a therapeutic area ever reported. And that doesn't matter what the virus is. The only virus that there seems to be a little question mark beside is being adenovirus in certain cases. But it's got a liver tropism, and honestly, most of the events you can look at are due to liver tropism. And you're kind of going, it's proven. We were very cautious as a whole field, all of us. When you look at the pox virus work, you look at the measles virus work, you look at our, adeno, reo, herpes. We've all been very cautious safety-wise, and what we've come up with is an area safety package, if you want to think of it that way, that is probably the best of any therapeutic area I've ever seen. It makes antibodies look horrid. Of course, it makes all low-line chemo awful. It's really, with some very, very minor exceptions that you just have to watch out for, really safe.
So how many of those 15,000 people, of the 15,000 people that have received this kind of therapy, how many of them have gone home and never had to go back to the hospital?
In every one of our studies, we always have standout patients who are supposed to be dead that were dying when they came into the study in the early studies that would be tumor-free and go home and like our very first clinical study, which we started in 1999, early 2000. I have a non-AIDS composed patient, last legs, probably 6 weeks left of life, who comes in every probably 6 or 8 months and strips down his underwear in my office. So I take pictures of him, gentlemen. But every one of ours has that. Our first glio study, we have a long-term survivor off of that was supposed to be always. I mean, but when you take people who are literally, the 9 toes in the grave and turn them around and and they're fine. It's very encouraging. And you just have, and we all have those experiences in this field. I mean, Oncolytics isn't alone in this. I mean, I'm very familiar with most of my colleagues' studies, and there's a lot of people, I think, wandering around today, even from early stage studies, that are doing very well. Now, of course, the proof is what BioVex is doing in phase 3s and what we're doing in phase 3s right now at the registration level, which is big randomized studies, control arms, the whole 9 yards. Yep. And we'll see.
So have you started a phase 3 yet, or they're just beginning?
We're about 8 months actively into our head and neck phase 3.
Okay.
It's in just under 30 sites now in 5 countries, and that'll be in close to 100 sites in about 4 months.
How many patients are enrolled in that?
I'm not saying exactly how many to date. It's a 2-part study.
And this will end up being able to use reovirus for head and neck tumors, right?
Yeah, it's second lines. So it's platinum refractory, taxane naive patients, and it's all metastatic disease. Like every one of these patients has metastatic.
And is it just virus, or is there another? It's not monotherapy, right? It's something.
No, it's carboplatin, paclitaxel, and Reo together.
Okay.
So CTR is what the investigators call it now. So
what about breast cancer?
We've had, I've had a couple of nice durable partial responses with Reo as a monotherapy in breast cancer, and they also took care of the liver mets in those breast cancer patients. Same with melanoma patients as a monotherapy. You've had a few like that. That it's interesting when you dig into the clinical data, and of course the scientists among us all do this, and the doctors start glazing over. But I love digging through clinical data, and I just expected we would see differential responses in metastatic disease, and of course nobody believed that when we started, because it's kind of heresy in oncology that you'd have something that would work better in mets than you would in primaries. And we're seeing, if you look across our studies, of about a 40% true clinical response rate in metastatic disease across studies, and about an 80% stable disease or better rate in metastatic disease. Wow. And that's lymph, lung, and liver. Now, and liver seems to be the most aggressive. Now, we all exclude brain mets from all these studies because they're just the wild card, and they really mess up lifespan measurements. But that would be the gold standard. I mean, if you could do brain mets, that would be, that there's almost 300,000 people die every year in the US from brain metastasis. So, we're actually selecting for brain mets because most of the drugs that we use don't cross the blood-brain barrier. So patients that would have died of something else are now dying of brain mets. So, I mean, to have an agent that's that active against metastatic disease is interesting in itself. Of course, until a couple of years ago, metastatic endpoints weren't accepted by the FDA as an endpoint. It wasn't a disease. It was an outcome of some other disease. But there's been a couple of companies now that have gotten SPAs for metastatic liver disease that are measured separately from the primaries. And so we're wrestling with how to handle one of that in one of our studies, because I think the preponderance of liver responses in particular with reovirus is quite overwhelming. It's just really unusual. And normally you see low single-digit response rates in that, and we're taking that up to 40, and that's a big difference. And they're durable too. They're quite long lasting, and they last for a long time after you quit treating. I mean, they do regress eventually, but it's quite remarkable. It's kind of neat.
How do you? So do you expect after this phase 3 that you will be able to go to market with a reovirus for head and neck tumors? Is that
yeah, absolutely. This study was done under a special protocol assessment. So the FDA, to the degree that you can hold them to that, signed off on it before we did the study, and so this will count as a single registration study, and they've already said that it will count as a single study.
Mm-hmm.
So are there other viruses out there now that are now being used that you're going to piggyback onto, or are you going to be the first?
Well, the only approved virus are in China. The old 015 vector from Onyx got sold by Onyx to a Chinese company who then got it approved in China, and they have limited sales there. So that's the only jurisdiction that I'm aware of. People are legally selling virus. There are a couple of jurisdictions where people are illegally selling virus, but that's the only place, the only oncology use. So in the States, if you're looking at local therapy, so directly into the tumors, then BioVex certainly will be the first, I think, across the finish line there.
What virus are they using?
They're using an engineered herpes virus. It's got I'm sorry, going to the technical speaker, it's got a 34.5 deletion to take the neural toxicity part out. And then they use that as an insertion site for a CG, I think it's CGSF. So it's really an immune therapy. You put the virus in, and then it basically expresses that, and then you attract in.
So it doesn't actually kill tumor cells. It enhances the immune response against them.
Exactly. Yeah, they're using it as a payload delivery vehicle more than anything else, and that seems to work quite nicely. Actually, there's Generex is doing the same thing, and a couple other people are doing that same approach.
Yeah, the whole cancer vaccine field is really heated up, and that general virological approach is something that people have been talking about for a while in concert with that.
So you mentioned a lot of studies in various rodents, in primates. You mentioned GMP production of the virus. Was that all done at your company, or was some of it done elsewhere?
Well, we went for our research. We went for a kind of distributed model where instead of us setting up wet labs and trying to recruit everybody, we just went out and set up paid collaborations with people in universities and research institutes and hospitals. And we've normally had 10 to 15 of those running simultaneously for the last 10 years. So I think, and we've sponsored a lot of basic research, and our way of doing it is just to basically just say, here's the general area. Could you work in it and let people loose on it? Which is, academics love that, and demand that they publish, which it makes us a little atypical. And that's, I think, we've been able to work with very good people by doing it that way. It's also very cost effective for the company.
So, what are your funding sources?
We've been public for quite some time. We've raised around $190 million now over the history of the company.
That's pretty good. Even in Canadian dollars, that's pretty good.
Even in American dollars these days.
Well, that's why I said that, because I used to.
Yeah. I'm still getting used to the fact that the American dollar is less than the Canadian dollar.
We're not getting used to that, by the
way. Sadly, I think.
What's the size of your company? How many scientists are working there?
We have 19 people total as internal hires, and there's probably what I would consider 5 or 6 of us to be scientists, and I still have myself in that category. So we did the research out of house, with input from us. There's a lot of cross-publication there. The manufacturing, I've got whole scale manufacturing, and Matt Coffey, who's my chief operating officer, does now, and so him and I have directed a lot of that. But we again did it at out facilities that you only did for a couple of years, and so what we've done is just had what's in-house, what was necessary to actually run programs, and so we haven't done a lot of stuff specifically in-house from a wet lab perspective. It's been, you just find the best people outside of that place and contract and work with them. And of course, clinical studies are always run that way. So most of the people that work for me now are ClinReg people working on our clinical programs, honestly.
So now what's the plan, assuming the phase 3 goes well and you get the approval? Are you then going to build your own sales force for this whole thing, or are you planning on collaborating with that too?
Well, that's come down to just a number issue. People used to partner for soft issues, and soft issues being things like you didn't have the people or the money to do the things that big pharma could do for you. And that went all the way through running, big complicated clinical studies, paying for them and running them, and the sales and marketing side. And so you always, when you were kind of evaluating whether you should run by yourself or partner, you always had to factor in they're going to do it faster than you. They'll have access to more capital than you. They'll do it better than you. And that was the reason you could justify doing partnerships, honestly. They've downsized so much in the last few years that there is this massive pool of unbelievably skilled, qualified, experienced people out there that can do all those things. So then it comes down to, well, can you access capital? And so really, whether you partner or not turns into a access to capital question. So if the cost of capital is cheaper for me to do a public financing, which I've done lots of, then you keep running by yourself. If the cost of capital gets more expensive than the partnership, then you do the partnership. And that's what it's really come down to. And right now we're just running by ourselves. We have, most of the big pharma franchises that have oncology are looking at us and other companies in this area with a high degree of interest. And so I couldn't predict, honestly, today whether we'll be partnered or not partnered. And, it's an interesting process, but it's certainly in the end, because what we're here for is our shareholders, and in the end, what they're interested in is a stock price that's X higher, not half of X higher. And any decisions that we make are based on what the eventual outcome and the effects on share price are. And that's right. As a company, that's how it has to be. Right.
The history of all of these biotech companies, and not including yours, of course, but a lot of them end up as subdivisions of very large companies.
Tend to get phagocytosed, yes.
Yeah, exactly. Engulfed and devoured. But the lysosomal enzymes don't apparently work, so some of the personnel is still kept, but the facilities now become known as, like Merck or Pfizer or these large pharmaceuticals. What is your hopes and expectations for your own company in, let's say, 10 years from now? How would you like to look?
Well, if you're looking again from a shareholder perspective,
Sorry, I'm laughing. I talk to people all the time who can't quite answer questions like that.
That's you got to ask the question, though, yes, Alan. Yes. Come on.
Well, when we set this up, we tried to align our management's self-interest with our shareholders' self-interest to the greatest degree possible. So what we've always said is what will be the best for our shareholders will be the best for us, right. Which means we're incentivized from a stock perspective more than we are from a drawing salary perspective. And so, I'm obsessed with end game and what that means for share price. That's really, that means something to me personally because we've set it up that way. I'm the one who set it up. So, I was setting it up to make sure that it worked this way, and it has. But when you look at companies that are bought, basically, our friends at Cephalon are experiencing this right now. You had a company that was trading $55, $60 or whatever it was. Forecast, because their first product is coming off patent, so their royalty stream would disappear, probably dropping down to $30 or $40 next year. And all of a sudden, a bigger company comes popping over the fence and hits them with a pretty decent premium, $73 a share, and they're trading at 76 because everybody thinks it's going to take another $3 to get them, and blah 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 blah. That end game is a very nice end game for companies. So being phagocytized for the right price is a very good end game. Our friends at BioVex, I think, are very much would agree with that completely. Their shareholders, of which many are my shareholders too, are extremely happy with that endpoint. They got $425 million for the core company and another $575 million in milestones that will go to the shareholders based on reaching those milestones. Their shareholders think that's a great deal. Their management is tied into that upside, so they think it's a great deal. And so I think for us, if we're phagocytized in a couple or 3 years, I think that's probably a pretty good outcome.
So who taught you all of this? You're talking to scientists and lay people out there. How did you learn all this stuff? Because I've been around the block a few times, and this is a language that most graduates of science graduates, at least, haven't got a clue about. They discover something, and then someone comes along and teaches them all these things. How did you pick all this up? If you don't mind me asking, I mean, that's a personal question, but.
Well, yeah, no, I mean, my PhD is bacterial cell wall ultrastructure. Let's get really esoteric. There was like three people in the world knew what I was doing, and they all ended up being on my defense committee. And then we started collaborating with each other because nobody cared, right? I mean, I couldn't have picked a more interesting and irrelevant topic at the same time. Because I have my own EM and my own, literally I had my own.
Nice.
As a graduate student, I had my own thing. I just burrowed away in there. And my prof had 5-year grants, so we never had to write grant money.
Oh, that was great.
Life was amazing, and it was R.G. Murray, kind of the father of a lot of microbiology. He discovered the gram-negative cell walls had peptidoglycan. I mean, we're talking old stuff here.
Wow.
I had Carl Robinow down the hallway who discovered Giemsa staining. Phil Fitzjames who discovered parasporal crystals. Was
Robert
down the hallway.
Koch, I presume, was your senior advisor.
Yeah, pretty much. And fire had just become common for the use of cooking.
Well, in Canada, that's a recent invention. Come on, we know this.
And I walked backwards naked through blizzards to go to school every morning. So, yeah, it sounds like that. I'm getting a little long in the tooth for this conversation. But I think I had a really fundamental microbiology background. Didn't have a clue. Had really planned on being an academic.
Right.
Decided just almost on a lark to try applied microbiology, and went out and worked in the oil industry. So I was actually enhancing oil production in oil fields using microbiology, which works really well actually. And then I started doing like scale up and fermentation design, that kind of stuff. None of that prepares you for the business side of what I do now.
Yeah, exactly.
My job is
that didn't lubricate your entry into what you're doing now.
No, basically I had an event drop into my lap. There was a government research, a company that was owned by the government of Alberta called ChemBiomed, and the government had pumped, I don't know, $100 million or something into it, and they had a couple hundred people and 800 research projects, and it was your typical company, but not really a company. It was a research institute, and they did some amazing science, but the government just was tired of paying for them. So the minister of the department who ran that whatever at the time basically walked into my office where I was and dropped this in my lap and said, "Fix this. Your measure of success is I don't want to read about it in the newspaper."
Oh,
really? Oops.
You better buy the newspaper.
So, I buy a briefcase because I didn't own one, and I bought myself a cell phone because I didn't own one, and I marched across the field to their headquarters and marched in the door, and they had like 100 and some people working on them that day, and I fired 85 of them that morning. And they each got about 5 minutes to tell me what they were doing. This was all new to me. I didn't have a clue. And what we were left with was 6 or 7 really, really cool projects, like just amazing science. And so I said, okay, well, we don't want to pay for them, so we'll partner them. We partnered all of them except for one in 3 months.
Wow.
I thought partnering was easy in those days because it was for that, but that was the quality of the science. It wasn't me, honestly. So I was getting exposure to all this partnering stuff based on that. And there was the last project left over was just sitting there, and we decided to form a company around it. So I knew some guys in Calgary who do a lot of oil work here because I grew up with them. That's my friends, and they threw some seed capital at it, and we formed a company, and we went public almost immediately. And so I had to learn all this stuff on the fly.
Wow.
And here we are. That was in '94. I've raised probably, I don't know, $500 million plus for Canadian companies and different biotechnology companies now, and 4/5 of that with 2 companies, and the rest as a director on other companies. I mean, I'm on boards. I put on boards quite often in companies to take them public. That's kind of what I seem to be good at. My wife teases me. She goes, the thing you like least in the world is what you're best at. So you must have offended some minor deity somewhere. So, I am the biggest science nerd on the planet. I love science. I love research. I love all that stuff. I mean, my name's on most of the patents in the company. I'm involved with the research of the company, but my job every morning is to come up and service the public market and raise money so that Matt Coffey can spend it. That's the way we divide things around here.
Is the Heritage Foundation at all involved in any of this?
The Heritage Foundation was there on day 0. Or whatever they're calling it now. They renamed it something that nobody knows. And so we all call it the Heritage Foundation still. Right. It's something like Health Talks or Health is Good for You.
Linda Humphreys was a good friend of mine.
That's the one she's the one who phoned me up to talk to the guys at the University of Calgary.
She's a fantastic woman and a great fly fisher.
Absolutely, and just a super person, just a wonderful.
Totally agree. And her husband too, and he was in the oil business, as you know.
Yeah, and a darn good pilot, a very good pilot too.
Yeah, he's unfortunately not with us anymore.
Brad, can I ask you? You early on in this our conversation, you mentioned some other types of tumors aside from head and neck. So, do you have plans going forward to do some trials? I think you mentioned breast cancer and some brain cancers.
Yeah, we have a randomized study going right now with metastatic pancreatic cancer in the first line. That's an interesting study in that we're not using Gemzar. So we're taking patients who have never been treated and saying, please forego the standard of care, which is Gemzar, and going to this randomized study where you'll either get carboplatin paclitaxel as the control arm or carbotax reolysin in the test arm. And we should have a data readout on that, late this year, early next year. We're running a randomized ovarian study with paclitaxel. So taxane failed patients are then getting treated with paclitaxel or paclitaxel Reo. And we have good early clinical evidence in humans that would say that those two things should work. So that's why we're doing them. We have a number of single-arm studies, so without control arms, that will be candidates for randomized studies. This summer, we're running 2 lung studies. We're doing a squamous cell lung study, a phase 2, and a non-small cell lung that we're pre-screening for RAS status. Both those will have data readouts this summer, so that I'll pick one of those to do a randomized study later this year. And the last randomized study this year is likely to be colorectal. And it's kind of funny that colorectal will be the 5th randomized study. It should be the 1st. That's the native habitat.
Sure.
Whenever it metastasizes to the liver about 2/3 of the time, and so that'll be a randomized study later this year as well.
And that's given intravenously for the colorectal.
Everything's intravenous.
What about cervical cancer? I didn't hear that mentioned.
Well, cervical cancer is interesting in that a subset of cervical is squamous.
Right.
And the non-HPV cervical cancers are mostly squamous. And if you look at our most aggressive responses in head and neck, they're squamous cell. You look at our squamous cell lung responses that we've disclosed, very aggressive responses. And so the obvious next one is to look at squamous cell cervical cancer, because there seems to be a sweet spot there for Reo and squamous cell for some reason.
And bladder cancer?
We've only treated a few bladder cancer patients, and they've all been metastatic bladder cancer patients. Matt Coffey's father-in-law used to run the Tom Baker Cancer Center here, and he used to say, stay away from superficial, because everything works in superficial bladder cancer. I mean, you just need to irritate the lining of the bladder. He says if you instill whiskey, he's Scottish, instill whiskey into a bladder.
Which is easy for us Scots to do, actually.
Yeah, they just do it from the other end. I was thinking catheter, and he was like, no, Brad, I don't meet with a catheter. But,
no. We do it the old-fashioned way.
Yeah, I don't drink whiskey, so I was thinking of it medically, not medicinally. But we know we've had some really nice responses with metastatic bladder cancer. But again, it's just, we're a little company and we don't have the resources to chase everything.
Well, I was just thinking of an overlap that occurs in Africa between urinary bladder schistosomes, schistosoma hematobium, because we have this other podcast called This Week in Parasitism. And there's a squamous cell epithelioma that develops in a lot of these patients.
Yes.
And there are co-carcinogens associated with it, and then that tends to metastasize, and then, of course, the patient dies. But I wondered how this would fit in with that, because you could probably just infuse it right into the bladder itself.
Oh, absolutely, and we've done animal models on that up at the U of A in the cross cancer. There's a couple of researchers up there that are interested in that. And if you give basically rodents metastatic bladder cancer and infuse it, directly into the bladder, and it works marvelously well. It's a great delivery system.
Very interesting.
So all of this is with reovirus. You have plans to do any other viruses, or is that?
We do, and there's a number of virus projects around that we're looking at. Our corporate strategy being get phase 3 data before we spread ourselves out into new areas. So as soon as we get phase 3 data, then I think you can expect us to be visibly looking at other viruses.
So soon Canada will be cancer-free, is what you're trying to say.
Well, right now, I'm looking at somebody walking down the street smoking. There's always new patients.
Yeah, that's true.
Carry on smoking, young man. So
the sad
fact is that many of the cancers we are trying to treat are environmental.
Sure.
Most head and neck cancers are environmentally caused.
Yep.
With exception, HPV exposure is environmental too. But if you don't smoke and you don't hang your head over a charcoal fire, which we do voluntarily in North America and not so voluntarily in places like India, or chew betel nut, which is a hugely aggressive carcinogen,
isn't that interesting?
You don't get head and neck cancer. So, we could eliminate head and neck cancer by changing, habits. It's like type 2 diabetes. It's a self-induced disease.
Yep.
I think a lot of our cancers in the end are, lung cancer is a classic. Most bladder cancers are due to smoking.
Sure.
People don't think about what smoking does to the human body. It's,
That's right.
so we could probably cut the cancer rate in half just by changing people's personal habits. That's not going to happen. Yep. People are not going to change their habits when they're 20 for something that's going to bother them when they're 70.
How about mesotheliomas?
We've treated only one mesothelioma and had a really good response, which means, it's one data point, so it doesn't mean anything. But it's better than no
so that's
response.
100%.
Yeah,
I'd call it 1,000%.
I could file for product approval.
That's right.
That's right.
So, Brad,
he did really well.
I have one email that I want you to listen to and perhaps answer, and then we'll let you go, okay? And this one is from a listener, Ivan, who writes, "Hello, TWiV team. I'm an avid listener to this show, TWIP and now TWIM, and this will be the second time I wrote with a question. We all know about the use of lytic viruses in the treatment of cancers, which can be very promising. I was thinking if there could be another way to use viruses in cancer treatment, if the tumor cells are infected with a modified virus that will promote overexpression of MHC class I proteins, then hypothetically the immune system would be stimulated to get rid of the infected cells by CD8 cytotoxicity, NK cells overexpressed MHC class I, and by the natural proliferation of the virus. I was just wondering if such a treatment is possible. I haven't seen any research paper describing such a methodology, and my knowledge is still not sufficient enough in order to answer this question myself. Keep up the good work, and I'm looking forward to many future episodes of TWiV. Ivan is from the UK, part-time biomedical student and electrician, and hopefully future virologist." So what do you think, Brad?
I will go one level up and answer that question. When you look at viruses for treatment of cancer, you really have three categories. The first is as pure vaccines, where you're basically infecting a tumor some way and causing a pure, like what I think of as a vaccine-like immune response against the tumor. And that's that technique's been tried and hasn't worked all that well, candidly. But I think it's more because as we all know with immune therapies, it's a little tough measuring. And now that we've got the Dendrion example, we can actually measure things. The second is the payload delivery vehicle method, which is the herpes story with BioVex is a good example, where they're using a delivery vehicle and basically are putting something. And that something could be what they do or a variety of other things. I can think of 8 or 10 different cytokines you could probably do the same thing with. And they attract in an immune response, but it's not a vaccine-like immune response. It's more it's more of an infection-based immune response, if you want to think of it that way. And the third is what ours is, which is using an active oncolytic virus that just is the cytotoxin itself. And they all kind of a little bit of overlap. Of course, Reo has a secondary vaccine-like effect after the fact, and so on. The approach that the emailer did, I haven't heard of people doing, but it is, I think, perfectly logical and perfectly feasible. Somebody could do exactly that, and it certainly would fit in with that. That's more probably the second category with the payload delivery. But again, you get a little crossover. I think the immune area is once we figure out how to measure things properly, and of course we're stuck with basically overall survival as really the only effective way, because you don't really see responses as an early indicator in the traditional way, and part of that is because it's kinder and gentler if you think of it that way. I mean, you look at panc's probably the best example of that. It's a pancreatic tumor and a primary tumor is 80% normal cells. If I get rid of the other 20% and the tumor shrinks 10 or 15%, it's not even called a clinical response. But if you look at the neoplastic tissue, it's gone. So we're used to cytotoxins that just trash the whole tumor. So if you take these nice gentle immune things and they get little, they stabilize the tumor, and the tumor might shrink a little bit and it's graded as stable disease at best, you may very well have gotten rid of all the neoplastic tissue. And where you're only going to see it is when you look at lifespan, and that's with Dendreon. I mean, there's no early benefit that you see with Dendreon's product, but they get lifespan, and that's what counts in our business. So I think that email approach is hugely doable.
Wow.
And I would encourage that person to pursue that thinking. So.
Yeah.
Well, Brad, I hope you are wildly successful for the sake of all cancer patients out there.
Yes.
And that's something that people tend to forget in our business too. I mean, we get obsessed with, my earlier talk, which was, shareholder this and shareholder that, is that I don't know a single person in any one of these companies that, if you scratch the surface to different degrees, doesn't come to what we're talking about right now, which is, any one of these products, when they're successful, will make a huge, huge impact on the quality of life of possibly millions of people. That's pretty impressive. Right. And I'm hoping it's us, but if it's not me, I hope it's one of my colleague companies.
Yeah.
You sound like you're well positioned. So
I got my fingers crossed. We're at the fun stage. This is the we're not talking 10 years down the road. We're talking, in the next year or two, we're going to know.
Great. Looking forward to it. Well, thanks so much for joining us, Brad. We really appreciate it.
Thank you.
Yeah, we do.
Well, I appreciate the time as well.
All right. Bye-bye.
Take care.
Brad Thompson, CEO of Oncolytics Biotech. Wow, 1 year. That would be great.
We'll have him back.
Yeah.
Well, he'll be too big to talk to us then.
Oh, no. He might want to fund us. Please, don't pass up that opportunity.
He sounds like he's good at raising money, Dickson.
Absolutely.
All right, let's do a few more email here. The next one is from John. He writes, I like the picture with the TWiV 120 sorry, let me start again.
Please.
Like the picture of TWiV 124 with the rabbits. The picture brought back lots of memories. It's featured in The Web of Life, a biology textbook in Australia in the 1970s. It was purported to be taken on Wardang Island, near where I first taught. A small island just off the coast, rabbits had been released onto it with none of the control measures. Water was extremely limiting, especially in the summer, hence the concentration of rabbits. I think I put up a picture for that episode, which was Grant McFadden, his anti-tumor viruses of rabbits crowding around a waterhole. That's what he's talking about. It also featured later in the rabbit control story in Australia. In the late 80s, trials were run there with another potential disease control measure, calicivirus. It was evidently working well, however, and before an official release, it escaped and did considerable damage to rabbit numbers. I remember hearing that the suspected culprit for aiding and abetting the escape were the local blowflies. You know about blowflies, Dickson?
I do, actually. They're carrion feeders. They lay their eggs on dead things.
It sounds like they picked up the calices.
Yes, so they had carrion luggage.
They did. They did. Well, they got rabid and decided to forget about them. So.
Thanks for the podcast. Gives me both understanding and a few stories with which to embellish my secondary school teaching. Very good. Eric writes, hello, disembodied voices that come from my iPod while I am passaging my cells. We're disembodied, Dickson and Alan.
I certainly feel that way. I don't know about you.
Recently, TWiV has discussed online resources for researchers. The conversation made me think of a very interesting group of sites called the Stack Exchange. The basic design of these sites is to provide a free place for questions on a variety of subjects to be answered by people in the know. It all started with code monkeys who wanted to build a site where programmers could ask questions and get useful, well-thought-out answers from other programmers. So, the site was designed well. The idea was so successful that they've adapted their site to be used by many other disciplines. The brilliant kick is that it's not a forum. The community votes on the answers given to questions. Those answers with the highest scores appear first and foremost underneath the question itself. Also, community members gain reputation based upon their activity on the site and how well their answers are received. So in a way, answers to questions are peer-reviewed. There is a biology site that is currently in the proposal stage, meaning its ultimate scope has not been fully fleshed out. It needs people to follow the site and mold the final product by suggesting on- and off-topic questions and rating those that are already there. From what I've seen from reading the example questions, the site would focus on technical questions pertaining to research. Stack Exchange wants specific questions with specific answers. Interesting note, one of the proposed questions was, is a virus a living being or not? Strangely,
well, that's not very specific.
If they had just left out the or not, it would have been.
Strangely, the question hasn't been shot down yet, despite not having a concrete answer. encourage people who want to see this site, gives a link for it. I guess I should end up by updating TWiV. I'm a graduate student in microbiology, and I found TWiV and virology blog to be an incredible resource for my development as a scientist. I'm also broadening my horizons by listening to TWIP and TWIM. You are all inspirations. Plus, I got a Drobo out of it, so that's cool.
Hey, there you go.
All right. Alan, you've been on some of these similar sites, haven't you?
Yeah. So when I read this, and he has a follow-up email saying that they have actually, apparently they've committed to launch this biology site, and so they're looking to generate some traffic to it. And initially I thought immediately of a site that works exactly the same way called Yahoo Answers. And uses precisely the same model. There's community voting on the quality of the answer, and then the winning answer, either chosen by the person who asked the question or by the wisdom of the crowd, ends up floating to the top. And Yahoo Answers has definitively and repeatedly disproven the old adage that there's no such thing as a stupid question. So there is a danger in this approach for anybody who wants to see the possible outcome. I would just Google the phrase "How is Babby formed?" That's Babby, B-A-B-B-Y, and you'll come across a video and that was derived from a Yahoo Answers discussion. Now, if this site is set up to kind of guard against that sort of outcome, then it might have some potential. But it's just a cautionary tale that when you take a majority vote, you don't always get the right answer. And I won't extend any political lessons from that, but people can just ponder it.
Voting as to whether something is correct or not is kind of bizarre.
It's certainly not a scientific approach.
No. It certainly ain't.
No, here we don't need consensus.
You could be the only right person, and everybody else could be wrong, and you could still ultimately be right. So,
the history of science is full of people who one person found something out and was right, and nobody else believed it until they proved it.
Exactly. Yeah, we don't need consensus. We just need innovation.
That's right.
Okay, the next one's from Jose. Hi, TWiV crew. I'm Jose, and I'm still a PhD student in Mexico working with innate immunity against dengue virus. This is my second letter to the show, and I'm very thankful for the comments to my doubts in the first letter, TWiV 82. I like the repeat questions. It's good.
Absolutely.
I'm still a big fan of TWiV since the early episodes. My letter is about some thoughts about this miRNA stuff. You guys gave a wonderful analysis of herpes virus Cimeri paper, where I hear that Dr. Racaniello mentioned a short comment about the hepatitis C virus history. I forgot all about that until now. I'm at the NIH making some collaboration experiments at Dr. Whitehead's lab, where a postdoc in the lab next door apparently wants to use this microRNA mechanism in a vaccine against West Nile virus, directing the viral RNA to degradation and maybe changing the neurotropism. Awesome, right? I just read the science paper that shows miR-122 as a cellular factor needed for the HCV replication. It's a very interesting paper and makes sense with the cell type restriction of Hep C. They found two possible complementary sequences, one in the 5' and the other in the 3', and apparently the one in the 3' end is less important for the accumulation of viral RNA. I just remember that RIG-I ligand in the cell line HUH 7.5 is supposed to be in the 3' untranslated region close to this complementary sequence to miR-122, and it's sequence specific. Do you think that the recognition of the Hep C genome by RIG-I might be the complex between miR-122 and this sequence? So miR-122 is a liver-specific microRNA that is required for efficient Hep C replication. And he's talking about where the target for miR-122 is in the genome. Maybe that's why the recognition of RNA by RIG-I in a sequence-specific manner only has been clearly described in Hep C infection of hepatic cells. In the paper where the PUUC sequence is suggested as the ligand of RIG-I, HUH 7.5 cells were used. The structure formed by miR-122 in the Hep C 5' UTR may be a better ligand for RIG-I. In the paper, it's suggested that the inactivation of miR-122 didn't affect the total protein synthesis, suggesting there is no effect on cell response. However, I would like to see the protein synthesis when the complex between the viral genome and miR-122 is in the cytoplasm. I found another related paper that shows interferon and ribavirin doesn't increase the microRNA concentration, but they didn't really measure the recognition of the genome. We really don't know if the recognition of viral RNA by RIG-I might be increased in the complex with miR-122. Thank you. Keep the wonderful job, and sorry my English still crappy. It's not crappy at all. It's great.
No, it's better. It's better than my Spanish.
I was just going to say.
So
it's
I
better
asked,
than your Spanish. No, I was going to say it's
I asked our friend Matt Evans, who was on TWiV some time ago to talk about Hep C, what he thought. And he said, since Peter Sarno's lab first showed that miR-122 directly interacts with the Hep C genome to stimulate replication, a very important question has been how this effect is actually mediated. In their original paper in Science, they showed that translation was not generally stimulated by miR-122 binding, and after some controversy, this has seemed to pan out. Instead, miR-122 seems to influence either Hep C RNA replication or stability. In fact, a recent paper from the Sarno lab, Macklin et al., PNAS 2001, has provided some of the keenest insight into this subject. miR-122 doesn't only bind with its short seed sequence to the two targets near the 5' end of the plus genome strand, but also makes additional contacts at the very 5' uncapped end. They speculated that this may shield the end from nucleases or prevent its detection by RIG-I. Thus, I think the actual effect may be the opposite of that suggested by this TWiV listener. miR-122 binding to the 5' end of the genome may actually shield the 5' triphosphate from RIG-I surveillance. Sure, the poly-UC track and the 3' end can strongly stimulate the RIG-I response, but the 5' end may also contribute to this effect. This is probably being furiously investigated in many labs right now. So RIG-I recognizes RNA with a 5' phosphate. So he's suggesting that miR-122 binding might mask that and hide it from the innate immune system. That's interesting.
Yeah.
All right, thanks for that, Matt. And let's do one more from Christina. I'm a neurograd student, and I love TWiV. I only wish there were a neuroscience equivalent. Maybe someday you will make twin. I don't want twins.
No.
I already have twins.
That's right. You've got 3 kids. That's
I already
enough.
have twins. Just kidding. I'm writing because I was just thinking how much I loved math camp in high school and how many other people I know went to some kind of math-centric summer school. Neuroscience seems to also run lots of summer schools at Woods Hole or Cold Spring Harbor or elsewhere. Is there a virology equivalent?
Ooh.
I'd love to run a virology camp.
Summer camp.
Be great.
Yeah.
Nice.
I bet Rich Condit would join.
Absolutely.
That's an excellent idea.
I don't know of any, Christina, but it is a great idea. We could actually do phage experiments.
We could do.
Right?
Yeah.
Because you don't get sick. Or reovirus, because they don't make you sick either.
There you go.
Should I read one more, guys? What do you think?
Short one. Pick a short one.
Well, that's a long one.
Pick us a short one.
Okay, one short one. Here you go. This is from Timothy. I'm an avid listener to TWiV, the one who listens at 2x speed. I just wanted to let you know that you inspired me to create my own podcast.
Oh.
I hope you forgive the name similarity to your series, but it's so good I couldn't resist. We are calling it TWIPO for this week in pediatric oncology. I pay homage to you in my opening monologue of the first episode, time 5:20. We just went live on iTunes today, and he gives us the link.
Hot darn.
Thanks again for all your work with TWiV, and TWIP. This is from Timothy Kripe, who's a MD and professor of pediatrics and oncology at the University of Cincinnati.
Nice.
A pediatric oncologist talking about what he knows. It's great.
Awesome. Excellent
Wonderful.
idea.
Absolutely wonderful.
And best of luck. I have listened to it. I even sent in a question.
Cool.
All right, let's do some picks.
Okie dokie.
Let's start with Alan.
Oh, my pick this week is a site that, well, if you read science, you probably heard of this already. It's a site called Science Buddies. And they just won a prize from the AAAS, so that's why you would have heard about it in science. They got the prize for online resources in education. This is a site that is full of ideas for science fair projects.
Aha.
And I picked, just a couple of weeks ago, I picked another site that's a little like this with a collaboration of researchers. This is actually set up so that it's just, there is a forum there's an Ask an Expert forum. But then there's, I think they have over 1,000 now project ideas. And so if you click on the project ideas for the site, you can look at them by category, or you can just browse through the directories. And if you pick a category, then you can choose what grade level you want. So, if your kid has a 3rd grade science fair project, you can calibrate the little slider at the top of the page and get 3rd grade appropriate projects in any of these different subject areas. And it's, a fair amount of the stuff is predictable, common science fair fare. But then also a lot of ideas that are coming directly from researchers who are working in these fields, who have done stuff that is easily ported to that environment. Just really, really targeting that audience. And of course, it's open for more scientists to participate. You can get into this thing, and if you've got project ideas that would be appropriate for kids to do as science fair projects, there are millions of kids out there who need these ideas, and you can write them up and present them to this site.
Cool. That's nice. I'm looking for virus projects, but it's mostly Meet the Science, Ask the Expert that I'm getting here.
Yeah, and under project ideas, if you go to microbiology, it's obviously you got to be working with stuff that's easy to grow and harmless.
Right. Cool. Very
and
nice.
Usually cheap.
Cheap.
Usually cheap. Cool. Very nice. All right. Dickson Despommier, what do you have for us, Dickson Despommier?
Well, if I haven't picked this one already, what episode are we up to?
131.
We could look back through the archives and see if we've ever picked this movie. It's called Insects and Angels, or Angels and Insects. Either way, I think it's Angels and Insects. But we could just check it right now.
No, you picked, Alan picked For the Love of Insects, but that's the only.
No, this
Angels.
is a different movie altogether. I don't know if Alan has seen this movie or not. It's it takes place at the time of Darwin in England, and it pits the heraldry. The reason why I'm picking it is, of course, that today is a very special day in England. It happens to mark the day of when Prince William has married a commoner. And the word commoner, I think we're all commoners under the skin, but it's very difficult to actually believe that if poor Charles Williams dies while he's in power, so to speak, as the king, this poor woman is dethroned as the queen. She does not become the queen, but somebody else in line for the next royal position gets the job instead. Okay, it's not a matter of just marrying into the family and then being part of it, although if you look back through the history of royal families, you'll find lots of aberrations. And this movie actually focuses in on the attitude during the turning point where Darwinian theory actually disproves hereditary yielding of power to the next level in the same family, and the ills of inbreeding. And what that brings to the world, and why we shouldn't be doing that. It's a fantastic movie, but it also looks at the evolution and behavior of ants. And you'll love this movie. I just predict that most of the listeners out there will just love this movie if they're not already familiar with it. So, I guess it's Insects and
Angels. It's Angels and Insects.
Angels and Insects, and it's a remarkably well done movie. The acting is uniformly great. And the theme is overpoweringly biological, and it's just a wonderful treatment of a scientific subject in the popular press that actually succeeded. So that's my pick.
Cool. Thank you, Dickson.
You're welcome.
Very nice.
You're welcome.
My pick is a Microbe World video, number 48.
Uh-huh.
It's called Emerging Diseases, the Importance of Early Warning and Surveillance Systems.
Cool.
And it's an interview conducted by Stan Malloy, who often comes on TWiV with me, and he interviews none other than Steve Morse.
Here you go. My co-leader in the course that we called Emerging Infections.
That's right. He's a professor here at the School of Public Health, right?
Yes, he is, of course.
And he's an expert on this sort of thing.
Yep.
Stan caught him at the AAAS meeting recently. It's about a 10-minute video interview.
Nice.
Check it out.
Nice.
Excellent.
I see Steve frequently, so there you go.
We both do.
And we also have a listener pick of the week. This one comes from Josh. Hello, Vincent, Rich, Allen, and Dickson. I would like to suggest Dr. Racaniello's Biology W3310 course, Virology, on iTunes University for the listener pick of the week. I've been listening to TWiV for about a year now and found his course on virology on iTunes U. I do not have a biology background or a science background, but have had no trouble following the virology course. If you love TWiV but never have had virology, this is for you. Here's the iTunes URL. Well, thank you, Josh. Keep up the great work, he says. And, well, it's not self-serving, is it, Dickson?
Of course not. But what if it was?
It's a listener pick.
It's a listener pick. And his arm is not actually broken. It's just sprained.
Huh?
No, that's
From patting his own back?
No, from twisting it.
I don't know what you're talking about, Dickson.
Nothing.
I'm going to cut it out. There you go. Another TWiV, Dickson and Allen. I hope Rich enjoys this while he's on the links. Speaking of Rich Condit, I just got a text message from him. I guess he's on the golf course. He says, TWiV newsflash. Condit leads with a 41 at the turn. All right. I guess we'll find out what he does at the end of it. Thanks, Rich. Of course, we are on iTunes. We're at twiv.tv, the Zune Marketplace, and microbeworld.org. And we also have an app for your Android or iPhone, microbeworld.org/app. If you are new to TWiV and you listen in iTunes, go write a review there. Just a short one. It'll help keep us on the front page so more people can check it out. Brad Thompson's at Oncolytics Biotech. Very interesting stuff. Hope it works.
Yep.
Alan Dove is at alandove.com. Thanks, Alan.
Always a pleasure.
Good to have you back. And Dickson Despommier is right here in my office. Thank you, Dickson.
My pleasure.
Verticalfarm.com. And I am Vincent Racaniello, and you're not. You know who used to do
Chevy
that?
Chase.
Chevy Chase.
That's right.
Yeah, we're of that generation.
We are.
I'm at virology.ws. And you have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.